First of all, let me introduce myself a bit. I'm Hiroto Takahashi from Osaka University, Japan. I'm a radiologist, and my subspecialty is neuroradiology. Neuroradiology is one part of radiological field covering imaging of mainly brain and spine. So basically, it's imaging for central nervous system. Diagnostic radiology refers to the field of medicine that uses non-invasive imaging scans to diagnose a patient. Diagnostic radiology can be used to identify a wide range of problems. Broken bones, heart conditions, blood clots, and gastrointestinal conditions are just a few of the problems that can be identified by diagnostic imaging. In addition to identifying problems, doctors can use diagnostic radiology to monitor how your body is responding to a current treatment. Diagnostic radiology can also screen for diseases such as breast cancer or colon cancer. As a subspecialty of radiology, neuroradiology can be used to help diagnose and treat brain, spine, head, and neck conditions. In this presentation, I'll talk about the brain imaging and research. Actually, magnetic resonance imaging is useful to get a variety of brain information, such as structure or its function. So let me move on to today's topics. First of all, I'll talk all about brain a bit, because brain is most important organ in human beings. Next, I'm going to refer what is MRI, magnetic resonance imaging, because as an imaging technique, MRI plays a major role in evaluating brain. Last, diffusion MRI and its usefulness for brain research using twin data is mentioned. So I'll start off with talking about brain. Even nowadays, brain still has many unknown things. The brain is the vital neurological organ composed of mainly cerebellum, brainstem, and cerebellum, and is housed in the skull and based in cerebrospinal fluid. Brain is consisted of gray matter and white matter. The gray matter is the substance of the brain and spinal cord that contains the neuronal cell bodies of the central nervous system. Within the cerebellum, the two main locations of gray matter are on the surface of gyri, cortical gray matter, and the nuclei of the basal ganglia. The white matter is the substance of the brain and spinal cord that contains the fiber tract of neuronal axons in the central nervous system. The term encases the axons in the tract compared to the gray matter. I'll show brain images of magnetic resonance imaging MRI. Nowadays, we can get the detailed brain anatomy information from MR images, and brain anatomy information from MR images can be used for brain research and clinical practicing, such as diagnosis of brain diseases or evaluation at the hospital. In brain, different areas of the cortex control different parts of the body. Each area has its own function. That is the localization of brain function. Brain areas have various functions for each. Let's see, such as thinking, planning, controlling emotions, movement, sensations, memorizing, hearing, understanding as a verb, and visually recognizing something. Illustration in the right side of this slide shows a cortical homunculus maps brain areas dedicated to sensory and motor processing for different anatomical divisions of the body. I'll introduce some representative functions of each brain area. Here we can see the frontal lobe colored area. Within the frontal lobe, of course, different area has different function. Executive functions such as thinking, planning, organizing, and problem solving, emotions and behavioral controls, and personality. Among these functions, one most important function is, I think, motor function from precentral gyrus. The parietal lobe also has a variety of functions depending on areas such as perception, making sense of the world, arithmetic, spelling, and most important function is, I think, perception from sensory cortex, that is from post-central gyrus. When it comes to the temporal lobe, most important function is considered to be memory from inside area that is known as hippocampus. Hippocampus plays an important role in doing information processing and the reproductive cycle, and is involved in Alzheimer's disease. The occipital lobe is located, as it's named, as the occipital area of brain. This area gets visual information from eyeball, and its important function is visual processing, so we can recognize surroundings. In brain, there are also numerous fibers connecting regions. They can be sorted as three kinds of fibers. They are commisural fiber, association fiber, and position fiber. Here I will show the corpus callosum connecting bilateral hemispheres, for example. The commisural fibers or transverse fibers are axons that connect the two hemispheres of brain. In contrast to commisural fibers, association fibers connect regions within the same hemisphere of brain and are categorized as short association fibers that connect adjacent gyri and long association fibers that make connections between more distant parts. Projection fibers of brain, also known as projection tract of brain, are a type of white matter tract that connects the cortex with other areas in the central nervous system, such as deep nuclei, brainstem, cerebrum, or spine. They may be efferent motor or afferent tract sensory. Here we can see one of the most important fibers, that is corticospinal tract, contributing motor function. Okay, in this section, I'll talk about MRI, magnetic resonance imaging. An MRI helps a doctor diagnose a disease or injury, and it can monitor how well we are doing with a treatment. MRI exam can be done on different parts of your body. It's especially useful for looking at soft tissues and central nervous system. There are both good and bad parts of MRI exam, as same as other exams. Good parts are no radiation exposure and high resolution in the tissue, especially in soft tissue or brain. By contrast, bad points are metal artifact, weakness to motion, and noise or long examination time. The MRI scanner is essentially a giant magnet. The strength of the magnet is measured in a unit called Tesla. Most MRI scanners used at the hospital and medical research clinics are 1.5 or 3 Tesla. MRI uses magnetic field and radio waves to measure how much water or hydrogen proton is in different tissues of the body and create images with water measurement. Then, MRI maps the location of water and then uses this information to generate a detailed image. The images are so detailed because our bodies are made up of around 65% water, so we have a lot of signal to measure. Here, let me show some representative brain MR images. They are called T2 weighted images, T1 weighted images, and FLAIR. MRI exam can obtain different types of brain images by running different types of MRI sequences. In the clinical practicing at the hospital, radiologists are using these types of MR images for evaluating diseases in brain. Here is a brain anatomy described with MRI. This is T2 weighted image of cerebrum. As you can see, MRI can describe the detailed brain anatomy. The cerebrum is consisted of such as cerebral hemispheres, basal ganglia, olfactory bulb, white matter tract, and corpus callosum. Brainstem and cerebellum are located under the tentorium. Anyway, brainstem plays an important role in the regulation of cardiac and respiratory function. It also regulates the central nervous system and is pivotal in maintaining consciousness and regulated the sleep cycle. On the other hand, the cerebellum has some features such as 3 surfaces, 3 fissures, 2 hemispheres, and single median vermis. Previously showed MR images are all actual images. Meanwhile, MRI can also obtain sagittal or coronal images and even oblique images as you want to see. Different types of view of MR images, such as sagittal images, can provide different types of anatomical information. Here is a different type of MR image that is called MR angiography. MR angiography can give the detailed anatomical information of brain artery. This is a case of acute brain infarction. In MR images, the white arrow points acute brain infarct area. Image A, that is called DWI, diffusion weighted image, can describe infarct area as high signal intensity clearly. Additionally, MR angiography can describe the disappearance of feeding artery. This is case of SAH, subarachnoid hemorrhage due to the rupture of brain arterial aneurysm. Subarachnoid hemorrhage can be seen as high signal intensity area spreading along with the brain surface pointed by blue arrows. Blue arrow also points brain aneurysm on MR angiography. Evaluation of brain tumor is important. There are 2 important cases that are primary brain tumors called malignant glioma with poor prognosis and metastatic brain tumor. This case has multiple lesions from lung cancer. MRI can clearly show the lesion like this. The roles of MRI are summarized here. Detailed information of brain anatomy from various MRI sequences can be given. MRI imaging technique is useful for assessing brain diseases in the clinical practicing. In this section is the most important part. I will talk about diffusion MRI or diffusion tensor imaging (DTI) and its clinical application for brain research in twin study. So first of all, what is diffusion and DWI? Diffusion weighted imaging (DWI) is a form of MR imaging based on measuring the random Brownian motion of water molecules within the voxel of tissue. In general simplified terms, high-risk cellular tissues or those with cellular swelling exhibit lower diffusion coefficient. Diffusion is particularly useful in tumor characterization and acute cerebral ischemia. So here is a case that is previously showed. In this case, DWI can show acute infarction clearly as marked high signal intensity area. This high intensity area reflects cellular swelling in this region because of cytotoxic cellular edema in acute brain ischemia, which leads restricted water diffusion in the intracellular space. Similar mechanisms can be seen in brain tumor with high cellularity. This case is lymphoma. This malignant brain tumor contains high cellular tissues, which leads restricted water diffusion in the intercellular space. Diffusion tensor imaging (DTI) is one of the most popular and widely used diffusion MRI techniques. By using DTI data, we can get the variety of information of brain neuron. DTI-based parametric maps can give the quantitative marker for assessing brain neuron. Tractography can visualize brain neuronal fibers. Diffusion tensor imaging (DTI) is a promising method for characterizing microstructural changes or differences with neuropathology and treatment. The diffusion tensor may be used to characterize the magnitude, the degree of anisotropy, and the orientation of directional diffusion. So basically, DTI can describe the detailed condition of brain neuronal fibers. Tractography, a non-invasive technique tracing brain pathways from DTI data, is increasingly being used for brain investigation. This technique could be useful to improve our knowledge about structural connectivity or to assess structural changes of white matter tracts potentially associated with neurodegenerative diseases. Tractography on DTI can describe the neuronal fiber in brain. This is the corticospinal tract motor neuron. So DTI-derived tractography gives detailed brain anatomical information and is used in the research and clinical practicing at the hospital. DTI-derived tractography can be used for the navigation in brain surgery like this. In case of acute infarction, brain neural damage is visualized. We can see diffusion tensor imaging based color maps of both healthy subject and a patient with amyotrophic lateral sclerosis, ALS. As white arrows pointed, the corticospinal tract function, that is motor neuron, appears thinner in the patient with ALS compared to healthy subject. From here, I'll talk about the utility of brain MRI technique in twin study. For the brain research, we have around 60 pair of twin study as East Asian data. Brain MRI data includes DTI to analyze brain neuronal fibers, and our twin data includes clinical data, genetic data as well. Regarding advantages of twin analysis, what can we get from twin data? In summary, comparing dizygotic and monozygotic data can reveal which or how much genetic or environment factors contribute to the phenotype, such as brain structure. Previous study with DTI reported there is an evidence that differences among individuals in white matter microstructure as measured with diffusion tensor imaging are under genetic control. Other study also mentioned that white matter microstructure is under strong genetic control. Anyway, now we are planning brain MRI research with twin data as follows, with a focus on the important brain regions such as motor neuron or hippocampus that have memory functions. We are going to evaluate which factor contributes to the region. I will introduce recent topic in radiology a bit. In the past decades, a field of medical image analysis has grown exponentially with an increased number of pattern recognition tools and an increase in dataset sizes. These advances have facilitated the development of processes for high-throughput extraction of quantitative features that result in the conversion of images into mineable data and the subsequent analysis of these data for decision support. This practice is termed radiomics or radiogenomics. This illustration shows the workflow of image analysis in radiomics or radiogenomics. This is the case of brain tumor. Throughout this image analysis process, many image characteristics are identified, which is data mining. Subsequent analysis is performed to correlate between various image characteristics and clinical or genetic factor candidate. With the use of the radiomics or radiogenomics technique, correlation with brain image characteristics and clinical or genetic factor candidate is performed to evaluate the relationship. So here is today's key takeaway. Diffusion tensor imaging can describe brain microstructure with tractography. In twin study, advanced image technique such as radiomics or radiogenomics may give the new knowledge about brain. Thank you for attention.